Hi, folks. This is Dr. Pickett. This is the slide narrative for Lecture 14, Chapter 24, which is the cancer biology chapter in your text. And again, I refer you to the PowerPoint, which you have access to as a member of my spring 2020 class. And I hope this kind of helps as we go through this. So what I'd like you to do, here we are starting at slide 1. I'd like you to transition to slide 2. This is just my favorite figure about biological information processing, where we kind of talk about how information retrieval from the DNA transitions into mRNA, which then is finally with a appropriately spliced and translatable mRNA is available to be translated into a protein. So with cancer, I just want to refer you back again to my earlier discussion in the semester of allelomorphy or allelism, because with cancer, we're going to need to explore thinking a little bit more about how specific types of different alleles can have an impact. Just as a reminder, if we have a mutation at the level of DNA, that mutation will, through the process of transcription, be faithfully transcribed into an aberrant mRNA, which then, through the process of translation, will be faithfully translated into an aberrant protein. So we're seeing a lot of quite fascinating alleles in cancer biology right now. Some of them are the classic amorphic alleles or complete loss of function alleles, depending on the literature you wish to refer to. These types of alleles, again, are deletions. If they are point mutations, they are point mutations in something like a promoter, or point mutations that cause a nonsense mutation in a codon such that very little or no protein can be translated from an mRNA. So corruption does flow just like information flows from the DNA to the RNA to the protein. And so we have to think about that phenomena of collinearity. Again, I stress the idea that, and my biochemistry colleagues might be irritated with me, but RNA polymerase, the ribosomes, these are dumb enzymes, dumb holoenzymes, dumb molecular machines. If there is an error in DNA, RNA polymerase will faithfully transcribe it into a corrupt mRNA. If there is a corrupt mRNA, the ribosome may faithfully translate it into an aberrant protein. If the corruption is so severe that transcription cannot occur, or no translation product can be made, that is a classic amorphic allele. And we see amorphic alleles in cancer biology. You do see situations where cells have acquired either through the germline, i.e. inherited from a sperm or an egg that made the zygote, or through what we call somatic mutation, they have acquired a mutation in a gene that is important to cell biology that is a complete loss of function amorphic allele. However, we also see a lot of alleles that we would describe, again, using our reference back to allelism and allelomorphy, as missense mutations, which cause a portion of the protein to not resemble the evolved and inherited form of the gene. You may have an amino terminus that looks very similar to the wild type allele, but then a carboxy terminus that is quite distinct because you've had a missense mutation, you've had a frame shift mutation, you've had another mutation that has either changed the identity of a single codon or may have changed the identity of literally all of the codons towards the carboxy terminus of the mRNA, the carboxyl terminus of the protein. So as we're thinking about cancer biology, we just need to refer a little bit back to thinking about the flow of genetic information from DNA ultimately to protein, and how if there is corruption in DNA, that will also faithfully flow through the system. A broken gene makes broken mRNA, makes broken protein if a protein is able to be translated from the mRNA. A change in DNA that results in a change in mRNA can also result in a change in amino acid sequence in the protein. So a long discussion of slide 2, but it's important for us to remember that given dumb enzymes, anything that's gone wrong in the DNA can result in an aberration in the mRNA and the accumulation of aberrant proteins. In cancer biology, we'll see that many of the alleles that we encounter, some will be complete loss of function or amorphic alleles. Some may be hypomorphic alleles, partial loss of function alleles. Some may be through missense. You may talk about them being a simultaneous loss of function. That protein's wild type function does not occur. But because, remember, I described proteins as Swiss Army knives, if there are some domains that are still intact, but other domains of the protein that are corrupted, those domains that are intact may be trying to function in the cell while the lost domains are lost. Those can be interpreted as loss of functions that are also coincident with something analogous to a neomorphic activity. That protein is doing something new in the cell now that it normally does not do with a wild type allele. So the whole zoo of alleles, which I discussed previously, we can encounter in cancer biology. Some of these alleles will be inherited from our parents. Some of the famous risks associated alleles with breast and ovarian cancer in women, or prostate cancer in some familial cases in men, or soft tissue sarcomas and other types of cancers. These can be alleles that we inherit from our parents, but also we will have many new alleles that develop because we believe because either they've encountered environmental insult, you've encountered something that is mutagenic in the environment, or simply because, as remember, I stressed the fact that many of the alleles that we see that are present in the panoply of alleles in a population often simply arise because DNA is soluble in water through the phenomena of isomerization, the addition or subtraction of atomic elements to individual nucleotides, and then the immortalization of those changes through DNA replication. So we chatted quite a bit about that when we were talking about alleles and mutation. In the context of this class, I don't talk a huge amount about the biochemistry of generating new alleles, the whole phenomena of chemical mutagenesis. I know I glossed that over fairly briefly. Certainly you'll encounter it more when you do nucleotide biochemistry in our biochemistry course. But for the context of thinking about genetic information, just understand that changes in nucleotides result in changes in the encoded form of genes in DNA, which will simply propagate through transcription and translation to mRNAs with sequences that are not wild type, and proteins with amino acid sequences that are not wild type, and that may result in a protein that is non-functional or has a function that is distinct, a gain of function or a neomorphy that may be distinct from the normal role of that protein. I remind you that, we talked about neomorphy. Sometimes those give rise to recessive alleles, sometimes dominant. We'll see that many of the alleles, when we talk about cancer biology, that are what we might describe as neomorphs or gain of functions of various sorts will end up being dominant alleles. Even in trans to a wild type allele, they will still confer a cancer phenotype to the cells and essentially the tumors that those cells will produce. Okay, so transitioning to slide 3, as I've stressed, cancer is a genetic disease. Many of these cancers arise as somatic cell mutations, even though there is some familial risk. It's surprising, but in many situations, the familial risk is a fairly low contributor to overall risk. In others, it's determinative. There are some mutations where if you inherit it from either your mother or your father, that mutation is simply determinative that your lifetime risk of cancer before 50 approaches the high 90 or even 100%. So some of these inherited alleles are quite dangerous. Some of them are less dangerous. Okay, so let's discuss section 24.1. When we talk about cancer being a genetic disease, what are the results of this corruption at the level of the gene as it progresses through? Well, not surprisingly, given our discussion of the regulation of the cell division cycle, we'll see that a lot of these mutations are affecting regulators of cell division. Why? One of the hallmarks of cancer, if you will, we discussed this very briefly previously, is unregulated cell proliferation, cell division, exiting from one mitosis, immediately entering into G1, immediately progressing through to the next mitosis. So mother cells make daughter cells, make granddaughter cells again and again and again. Now, because of this cell division process and the fact that it kind of starts with the accumulation of mutations in a mother cell that will eventually give rise to the entire tumor, we often describe tumorigenesis as a clonal process because a mutation arising in one cell is immortalized through DNA replication in the genomes of the daughter cell, which then again, through DNA replication and cell division is, and so tumors build over time. We often will describe this as building a tumor genome. We'll get into that in just a bit because one of the things we see is that micromutations, mutations affecting single nucleotides, single genes, and macromutations tend to accumulate over time as cancers develop. This is described as the cancer tumorigenic genotype or mutagenic genotype. And it simply means that tumor cells accumulate mutations over time. We'll explore why that is in just a bit. So unregulated cell proliferation is a hallmark of cancers. As cancers kind of increase in grade and become more and more severe in terms of their health outcomes, we will often see transitions from cancers being localized diseases to what's referred to as metastatic spread. That means that small clusters of cells from the tumor end up circulating either through the circulatory system or the lymphatic system, and they end up in parts of the body that are remote from the site where the tumor arose. Historically, that has meant that tissue of origin has been a very important concept for thinking about tumors. We are kind of in the modern era beginning to rethink the value of the tissue of origin. Does the fact that a tumor arose in liver, arose in hepatocytes, mean that that gives us deep insight into how to treat a tumor once it has become grave enough that it is presenting a risk to health? This now has become a more open question. Historically, there was a lot of correlation between tissue of origin and the treatment behavior that you would observe in physicians. Now the effectiveness of that approach and other ways of thinking about tumors, perhaps as unique and novel, almost life forms that have their own genotypes, and with their own set of genes, they must be approached in a very directed way based on the specific tumor genes that they have, regardless of the tissue of origin. That's kind of moving into the fore. Another thing we're starting to rethink at this point is whether or not localized disease, if you have a local tumor, you have not had metastatic spread, how dangerous, if you will, to life and health of the patient that is, and what the treatment approaches should be for a more localized tumor versus a tumor that's had metastatic spread. These are all discussions that are ongoing amongst oncologists. Dr. Pickett, because of his clinical literature courses that he teaches to graduate students, is developing a little bit of an interest in oncology. And as I've been studying it more myself, I've been fascinated by how much uncertainty there really is in oncology around many of the issues that I would say, even in classical oncology, going back to the 80s, I think were thought of as settled ideas, settled science. Now we're reopening a lot of that settled science and are finding it's a bit more unsettled than we thought. So unregulated cell proliferation and metastatic spread is something we see commonly. Moving on to slide 5, just as a reminder, what do we see kind of at the base of many mutations? Well, we do see biological phenomena like inflammation, other types of issues playing an important role. But by the time we're looking at a tumor that has transitioned from being a mother cell in a tissue, a hepatocyte, or a bone marrow cell, one of the hematopoietic stem cells, etc., to a cell that is contributing to a tumor, those cells have begun to build genotypes and genomes that are distinct in relation to the host. Mutations have changed alleles. Now, just as a reminder, what are the range of mutations that we see? They're quite dramatic. We may see single nucleotide substitutions, other single nucleotide or short stretches of nucleotide modifications. You will often see large-scale chromosomal rearrangements, amplifications, deletions, translocations, inversions. These become quite common. Aneuploidy, accessory chromosomes, all of the different types of mutated forms of chromosomes we've discussed previously. We do begin, at least in some tumors, to see these accumulate. Now, it's interesting because, for instance, if you compare many blood cancers with something like familial adenomatous polyposis coli, which is familial colon cancer, many of the blood cancers, you see a sort of weighting of smaller mutations, single nucleotide mutations, single genes affected, perhaps a smaller suite of genes impacted, whereas by the time you're kind of evaluating the genome of a adenocarcinoma from a patient with familial adenomatous polyposis coli, with familial colon cancer, there you may see a whole range of problems. You'll see aneuploidy, you'll see rearrangements, translocations, inversions, accessory chromosomes, all sorts of problems. So when we talk about the cancer genome, we are talking about the fact that the genome is distinct genotypically in relation to the wild type genome of the host body, but the range of diversion, or I won't say diversion, of difference from the host genomes and the tumor genomes can be very broad. In some tumors, there may be a few changes that are made. In other tumors, it looks like it's building the genome of a new organism. the numbers of chromosomes have changed. Chromosomes are missing. Chromosomes are duplicated. Chromosomes have many rearrangements, translocations, inversions, etc. on them. So your book provides some very nice examples of some very cool clinical samples of different karyotypes. Now, if we look at if we look at slide 6, we see a beautiful comparison of a wild type genome in the A frame of the image and a mutant cancer genome in the B frame. What are these images that we're looking at? So remember I talked about karyotypes as being metaphase chromosomes that have been stained and looked at usually from white blood cells, although when you're doing cancer karyotyping, you use cancer tissue. And then you look through a microscope at these highly condensed chromosomes, and you see chromosomes with 2 chromatids, and you see the characteristic G banding or other R banding or other types of banding patterns, depending on the technology you're using for visualizing chromosomes. In this image, we're using, I believe, based on what they look like. Unfortunately, the reference in your book is a little modest. But these appear to be what we refer to as sky or spectral karyotypes. When you do spectral karyotypes, you use very small probes to specific regions that are complementary to different parts of different chromosomes. And they have associated with them fluors, chemicals that are excitable by incoming light and then produce fluorescence at a specific wavelength. So when you're doing spectral karyotyping, you use sets of probes that are complementary to the X chromosome, the Y chromosome, chromosome 1, chromosome 2, etc. But each of those sets of probes has a slightly different chemical mix of fluors on it, with an appropriately prepared fluorescent microscope, one that is capable of detecting spectral fluorescence. You can assign a specific fluorescence signal to a chromosome. You then take those specific photons and use post-imaging analysis to assign something like a specific color to a fluorescence wavelength. So you might assign, visible to our eyes, the color red to a fluorescence wavelength emanating from the probes of the X chromosome. You might assign a false color green to the fluorescence signal coming off of chromosome 2. And so by doing this, you develop very powerful tools to identify individual chromosomes based on their fluorescent signal. And if you have any rearrangements, you can actually identify which parts in a translocation are from the X, which parts in the translocation are from chromosome 15 or chromosome 21, whatever the particular translocation-derived chromosome is. So looking at the A figure here, what are we seeing? We are seeing a wild type spectral karyotype from an XY male. You'll notice here the X is blue, the Y is this kind of light white silvery gray. And you'll see chromosome 1 has its false color signal of kind of like light beige. Chromosome 2, dark brown. Chromosome 3, light purple. And so as you move through the autosomes and the sex chromosomes, you can see that these chromosomes with their hybridized probe sets are emanating through fluorescence a particular signal which is detectable. Now, if we compare that to the B karyotype, where again we have spectral karyotyping. What are we seeing? Well, I direct your attention to chromosome 1, where you see 2 copies of chromosome 1 that are present, but you'll notice they're a composite of 2 colors. Now remember, these are metaphase chromosomes, so each individual chromosome that you see here is the composite of 2 sister chromatids that are very closely associated together because of the state of the cell division cycle that these cells were isolated in, that these chromosomes were isolated from. So you see 2 copies of chromosome 1. But this is interesting because both of these copies of chromosome 1 are derived from the same translocation chromosome. You'll see that on the Q arm of chromosome 1, which is mostly this off green color, this kind of mauve color, there's a little tiny brown nubbin at the end of both of those chromosome arms. So this indicates that both copies of chromosome 1 in this tumor cell are translocation-bearing chromosomes. Take a look at chromosome 2. What do we see? One of the homologs of chromosome 2 has a terminal deletion. The other copy of chromosome 2 has a translocation on, I believe that's also on the Q arm. So, this is quite fascinating. Here we're seeing in the chromosomes, the metaphase-arrested chromosomes of a tumor, we are seeing simply laid out in a karyotype the tumor genome with all of these macromutations. Chromosome 1, both copies translocated. Chromosome 2, 1 copy deleted, the other copy translocated. Chromosome 3, if we just move down the karyotype, 2 copies appear to be wild type. However, we are aneuploid, at least for the centromeric region of chromosome 3, because arrayed next to it, there is an additional accessory copy of chromosome 3 that is an obvious translocation. You see the pink of chromosome 3 attached with a blue little nubbin, and you also see that the P arm of chromosome 3 is deleted. Taking a look at some of these chromosomes as we move along the karyotype, we can see that this genome is getting quite creative with macromutations. Chromosome 4 has 1 homolog that appears to be normal. The other homolog is a multi-chimeric chromosome. You can see that the P arm is translocated. It appears to be from chromosome 13. The centromere is derived from chromosome 4. The Q arm has a bit of chromosome, I'm thinking 14 attached to it, but then the telomere is chromosome 4. So there is a internal interstitial deletion of most of the Q arm of chromosome 4 with a simultaneous replacement by translocation of another chromosome. So looking at this tumor genome, we're seeing a huge number of macromutations, translocations, inversions, multiply translocated chromosomes, accessory chromosomes. Here we are seeing a human genome interconverted through mutation into a tumor genome. If we were to map some of these translocation breakpoints or to sequence, what would we find? Many point mutations, many translocation fusion genes, some of which are likely contributing to this disease state. So tumors over time through this mutagenic phenotype are building genomes that are supporting the tumor phenotype. The tumor genotype correlates with the tumor phenotype, correlates with systemic cancer by the time it gets bad. Okay, so let's transition to slide 7. So your authors make the claim that most cancers are somatic. That is, they are derived from changes that occur within cells in a tissue as something like a stable muscle cell becomes a myosarcoma, or a stable germ cell in a gonad becomes a testicular cancer, or a stable hematopoietic stem cell slowly builds over time its tumor genome and eventually becomes a hematologic malignancy or a blood cancer. They do make the claim that about 1% of the cancer incidence in the broader population is due to the inheritance of a germline mutation, either in a gene that is responsible for monitoring cell division and is lost, so the cell loses that monitoring, or is the creation of a novel allele that promotes cell division. We'll talk about these distinctions in just a bit. And certainly some of these cancer predisposition genes are in the broader society, and occasionally at a rate claimed by your book at about 1%. I don't know how broadly these have really been surveyed, but from physicians I've talked to, from our former alumni, that is probably at least in the ballpark in terms of incidence. Your book also, I think, makes this interesting claim that cancers are rarely associated with mutations in single genes, but are often associated only with cancer genomes in which multiple mutations have accumulated. And that does seem to be fairly true. I don't know if I would go so far as to say rarely do you see single genes mutated. I don't know how broadly we've surveyed that, but it's certainly a reasonable claim. Thinking about the genes that are targeted, from everything we've discussed in this class, where do we see mutations? In a lot of the suspects we would anticipate. We see mutations in things like cellular functions, like affecting DNA repair, regulating cell division, regulating programmed cell death or apoptosis. We also see mutations in genes that affect cellular development or differentiation into tissue types. These are very common in the blood cancers, where you see that blood cells which are in the path to differentiating into mature forms of white blood cells instead kind of seem to be arrested and continue to proliferate as intermediates. So they have an inability to fully differentiate, to get locked into a developmental loop that involves cell division. Also, we see mutations in genes that are involving migratory behavior, how cells move within the body, and also cell-cell adhesion or cell-cell contacts. So your book then moves into a discussion of the sort of classical view of tumorigenesis and how we classify tumors based on different grades. I will say this is a rapidly evolving area. I think we think of many tumors as being benign, or tumors kind of transitioning to a greater grade. But whether or not there is the inevitable progression of a tumor, or whether or not tumors that we think are perhaps at an early grade, but they may have already released micrometastases. And so even though we only see a localized tumor, we're really dealing with systemic disease everywhere in the body. It's just we haven't detected those tumor foci yet. So, a tumor being benign or malignant, these are more sort of classical clinical definitions. As we learn more and more about tumor biology, we may be discovering that some tumors, which we may have historically thought of as very large, likely to be malignant and aggressive, will in fact clear on their own based on the host's immune system, whereas other tumors, which we may grade very early because they've already released micrometastases, we may need to approach those in a much more aggressive way than we would have historically, because in fact, the disease, although it appears localized, is really systemic. So benign tumors have historically been classified as very local, slow-growing. Usually they are encapsulated. They have connective tissue surrounding them. They have not destroyed all of the connective tissue around them. Oftentimes, if they are surgically resected, that results in a full resolution of the disease. There doesn't need to be any chemotherapy or other treatment. So benign tumors, if they are treated surgically rather than just medically, often result in a full resolution of disease. That is in contrast to malignant tumors, which have a tendency to produce metastases everywhere in the body. Disease is no longer localized. It is general and systemic throughout the body. Malignant tumors are associated with more life-threatening complications. And this has led to kind of a rule of thumb, depending on the oncologist you talk to, at least the youngsters that I've trained over the years that have come back and chatted with me over time about this. This has led to kind of the 1 to 2 kilogram hypothesis that you generally will not see greater than 1 to 2 kilograms. So if you've got a pound of tumor, you're a rare bird. Why? Because by the time tumor mass has grown that much, it has crushed an important nerve. It has occluded or compressed an important vessel, an important artery or vein. And so you have a tendency to not have tumors present in the form of a patient presenting with disease that are extremely large because of the havoc they tend to wreak on the overall body system. So malignant tumors, even in that case, may have sort of an upper growth limit. Why? Because usually the patient has died if the tumor has grown past a certain point, or the tumor burden of the whole body has grown past a certain amount. So I talked about the fact that you can have a mother cell. If we transition to slide 9, I've talked about the fact that we can have a mother cell that accumulates mutations, that then produces daughter cells with mutations. More mutations will begin to accumulate. Because of this phenomena, we do describe most tumors, certainly primary and secondary tumors, primary tumor sites and metastatic tumor sites, generally as clonal. Now, the important distinction to make about that is as tumors divide, different lineages of the tumor will likely accumulate different mutations. So sampling tumors at distant sites in the bodies, we will see that their genomes share a common origin. There was a tumor mother cell that gave rise to them, but now the genomes may be quite distinct, given hundreds of millions, perhaps, of rounds of cell division and cell replication. So tumors in different parts of the body may have different tumor genotypes. This raises a lot of complexity for things like detecting tumors in small metastases that are floating in the blood or the lymph. We don't know if any sample we take at a remote site or a circulating site in a tumor bears a lot in common with the tumor that is threatening the life of the patient. So one hazard about that is if I do a blood draw and detect using very sensitive equipment a particular tumor genome, and then design a treatment or therapy around that tumor, if that tumor genome I've detected is not characteristic of most of the tumor genome in the body, I might kill only a subset of the tumor cells and leave the rest of the tumors untouched. So this is an ongoing challenge, I would say, to precision medicine, is that we have tons of tools for precisely genotyping cells, but tumor biology might be fighting against us if we guess wrong and we do a workup of a clone of tumor cells that in fact represents a minority genome, if you will, in relation to most of the tumor that's present in the body. So that might mean that in order to do something like liquid biopsy, we would want to be pretty certain that we have detected some hundreds of, or evaluated some hundreds perhaps, of tumor genomes to be sure, if we are going to design a therapy around what we do by a liquid biopsy, sampling metastatic cells, to be sure that we're going to design a therapy that will be effective against the bulk of the tumor. So we've talked a fair amount at this point about some of the macromutations. Let's talk specifically, beginning in slide 10, about some of the specific translocations that we do see correlated with cancer. We're going to chat a little bit about Burkitt's lymphoma, where we see reciprocal translocations between chromosome 8 and 214 and chromosome 22. You will see in any individual patient's Burkitt's within their tumor genomes, a characteristic translocation, but a different Burkitt's patient may have a different translocation. Simply depends on which one arose during tumorigenesis in those patients. Another reason, and so because of that, that really supports this concept that tumors are clonal. If we see in distributed Burkitt's lymphoma in tumors in many parts of the body, the same translocation in every cell, if we see it's chromosome 8 translocated with 2 in one patient, in another patient across the tumors, we see chromosome 8 translocated with chromosome 14. That also gives us support for this concept of clonality. And we'll talk a little bit more about some of those specific translocation chromosomes in a bit. Because of this idea of clonality, this has contributed to the formation of what we call the cancer stem cell hypothesis, that in metastases, there are cells within it that have the ability to build tumors as kind of independent organisms anywhere they happen to lodge in the body. Where do we see distributed tumors away from the site, the primary site of tumor formation? We see them associated with capillary beds, where metastases get lodged within vessels that have a small diameter and then they grow in that point. So this is why we'll commonly find melanoma in the brain, because of all the capillary beds that are present in the brain. While we'll commonly find melanoma tumors, even though they arise in skin, we'll find them within the lungs because of all the capillary beds within the lungs. We will find them commonly within the liver because of all of the capillary beds in the liver. So as these metastases are floating through the body, once they get into a capillary bed where you have your little arterioles and venules interacting, once they get to an area where the diameter is so small, the metastases can no longer move through that region. They will begin to lodge there, and they will do cell division in that region. Now, this is very fortuitous. The capillary bed is already supplying or providing a localized blood supply. So a capillary bed is a great place to form a tumor, because that tumor is, through the process of what we refer to as angiogenesis, new vessel construction, going to need to tap in to the blood vessels of the patient in order to supply blood to the tumor to support its growth. Tumors that fail in angiogenesis generally die as very small local foci of cell growth. They get to around 1 centimeter inside, and then free diffusion of things like water, nutrients, and oxygen can no longer occur. So these little local tumor foci develop necrotic centers. They die in the middle. If they don't do angiogenesis, or if you can stop angiogenesis, that is a great way to stop tumor formation. So these cancer stem cells within metastases lodging within capillaries have within their tumor genome and their cancer cell type the ability to rebuild a tumor anywhere in the body. Now, differentiating quote unquote cancer stem cells or establishing cancer stem cell as a particular phenotype in relation to other tumor cells is somewhat controversial, but it's certainly been a fruitful target for research and is an interesting hypothesis that will continue to be explored over the life of your career as a scientist. So where have we begun to hypothesize that cancer stem cells exist? Well, if you look at slide 12, there are a wide number of different cancers where we think we've detected cells that have this unique capability to rebuild the tumor in contradistinction to other cells clonally derived with the cancer genome that seem to have a cell type that is less, shall we say, pluripotential, less capable of rebuilding whole tumors distally. Although, again, I would like to note that it is still somewhat controversial whether or not these cells are particularly novel in relation to any other cell with a tumor genome that we might find in the body. So transitioning to slide 13, I'd like to introduce the concept of oncogenesis, which is this slow progression of tumor formation. Why do we think that cancer is kind of a multi-step process? Well, we know with familial cancers like familial anematous polyposis coli, where we can go into a patient and we can sample colon polyps. I'm sorry, I'm trying to do 2 O words at the same time. Colon polyps at different stages in their growth, genotype them, and we can see that the genome is changing, and as the genome is changing, cell types are changing, the ability to be metastatic, transitioning from a velar adenoma to a carcinoma. We see how the genome is changing. We see how the cell types are changing associated with that. We see that polyps arise early, adenomas arise later, carcinomas arise latest. So this suggests that this is a time-dependent process and that the changes that are accumulating must occur over time in order to finally get to the point where you are a full-blown malignant cancer that has put the patient's life at risk. So related to that, what else do we see? We see that the incidence of most cancers rises exponentially with age. The older you get, the more cell division you've had in your body, the more error-prone DNA replication you've had, the more error-prone DNA repair you've had, the more opportunities you've had for mutations to accumulate so that a cancer genome develops within a given cell. Another thing that we see is that, for instance, with something like familial anematous polyposis coli, we see that many mutations in distinct genes are required. Even though you've inherited a risk-associated allele from your parent, you will have other mutations that must occur before you finally get to the point where it's malignant. since these mutations can only accumulate over time as DNA repair becomes more error prone, as translocations and other macromutations occur, as micromutations occur. We also believe that this is kind of a slow, time-dependent process. Now, in an individual that doesn't have a familial risk allele that's inherited, we still see that a number of mutations tend to be needed in distinct genes before we progress to full-blown malignancy. That has led to the multi-step or multi-hit hypothesis of cancer, that early mutations in single genes set up cellular systems so that they become more error tolerant. Becoming more error tolerant, more mutations accumulate, and thus as time goes on, genomes increase in their tumor likeness, and eventually you have a full tumor genome. So your authors on slide 14 provide an interesting, if you will, age versus incidence graph that talks about or charts how different cancers increase in time as a percentage of the population. So this is annual incidence per 10,000 males, and you'll notice on the left axis, it goes to an incidence of 1%. An incidence of 1 in 10,000 males up to an incidence of 510,000 males. You'll see, interestingly, some of the cancers that are very sensitive to environmental factors increasing mutation rates, like skin cancer, where we know that ultraviolet light coming into the skin that we expose ourselves to every day are bringing energetics and mutation into the cells in which they are modifying nucleotides directly. But we see that skin cancer, with its strong environmental component, can begin to be detectable at a rate of more than 1 in 10,000 in males between the ages of 20 and 30. Other cancers, for instance, prostate cancer, and the prostate is a dynamic organ. It is the organ that provides most of the secretory volume to semen, so they're producing a lot of the fluid components. The prostate does lots of mitosis. It increases in mass over time due to mitosis, and correlating with more mitosis, error-prone DNA synthesis, error-prone DNA repair, those prostate cells, simply due to the fact that it is a tissue that is dividing along with cell division in the skin, makes it at risk for undergoing oncogenesis, but that rate increases over time, and we don't really begin to see detectable levels of prostate cancer until men increase to the age of 50. Then between the ages of 50 and 80, prostate cancer increases dramatically. But what is in common with all these tissues? Prostate, stomach, skin, rectal, pancreas, and esophagus. All of these tissues are cells like epithelial cells that are always undergoing phenomena like inflammation, cell division, wounding, and wound healing. These all require kind of continuous activation, because skin cells are made in our deep layer of skin, but then skin cells slowly, as they progress towards the surface and are finally shed as dead skin cells, our skin is undergoing continuous mitosis, continuous DNA replication, error-prone DNA replication, error-prone DNA repair. So one thing you see in common with all of these tissues is they are tissues that are continuously replenished by cell division. They are also tissues in some cases that are particularly exposed to environmental insult, mutation, mutagenesis, simply based on their position. So with more mutations over time, with more cell division over time, your lifetime risk or your risk of having an oncogenic event increases simply with your age. So it's quite fascinating. You'll notice with all of these cancers, by the time we get to 80 years old, even esophagus, where the lining of your esophagus is undergoing continuing mitosis, by that point, on the low end of this cancer spectrum, we have 50 in 10,000 people affected. On the high end, prostate cancer, undetectable at the age before 50, very, very low rates of prostate cancer. By the time you're at 80, 500 men out of 10,000 present with prostate cancer, and many, many more men have benign prostate hyperplasia, BPH. So many, many, many other men will have pre-malignant, if you will, types of cell division issues. So another bit of evidence that supports this kind of multi-hit process is the fact that there have been some situations in history where there was a time point of intense exposure to mutagen, whether it was a chemical mutagen with some of these chemical plant explosions, spills of carcinogens that exposed a broad local population to mutagenesis, or something like war, where we think about the dropping of the atomic bombs in Japan. There we were correlated with a single event. Now, if a single mutagenic exposure was sufficient to cause cancer, any individuals that did not die from acute radiation or heat associated with something like an atomic bomb, any individuals exposed to sublethal acute radiation would be expected to develop cancer perhaps immediately if single mutations in cells were sufficient to cause cancer. However, what we saw at Hiroshima and Nagasaki, for instance, is that there was a 5 to 8 year period lag before leukemia rates, for instance, began to spike. So what does that suggest? That suggests that the environmental exposure led to the accumulation of initial mutations, but then only subsequent cell division. The mutator phenomena eventually constructed the tumorigenic or the tumor genome over a 5 to 8 year period. So leukemias did not immediately spike, but with the delay required for cell division, DNA replication, error-prone DNA replication, error-prone DNA repair, only with the slow accumulation over time of mutation were tumorigenic genomes built. The initial mutagenic exposure to radiation certainly potentiated the cancer risk and elevated it dramatically, but that risk was translated into actual clinical presentation of disease through long processes of cell division and the slow accumulation of more mutations. So we do think that cancer is a multi-step process. It develops slowly over time. It begins with mildly aberrant cells with a few mutations or inflammation, other processes that are driving mitosis, which again, simply by being dependent on DNA replication is, if you will, mutagenic. DNA simply sitting in water will change and isomerize, thus mutations will accumulate. So in slide 16, your authors introduce this concept that tumorigenesis is the result of one or more genetic alterations followed by subsequent alterations, and they describe this process of accumulation of mutations, building the tumor genome, and cell proliferation as tumorigenesis. They would then like to differentiate our modern understanding of these types of mutations into sort of distinct classes. They talk about the fact that because of this mutator phenotype, there will be driver mutations, key mutations in genes that cause those cells to become fit as tumor cells. Whereas other mutations that are just random, they happen to be at a translocation breakpoint, but they're not genes that promote cell division, or they happen to be mutated due to a single nucleotide or other mutation, but they don't actually cause an increase in the rate of cell proliferation. They describe these sort of central mutant alleles as driver mutations. They are playing an important role in the tumor genotype and in the tumor phenotype. Whereas other mutations, they call them passengers. This is one of the real challenges for your generation. If we are going to take a genome-wide approach to understanding the cancer genome, we will eventually have to find some way of filtering out driver mutations from passenger mutations and being able to characterize them appropriately, because the hypothesis is still biologically plausible, I would say, but still to be confirmed experimentally. The hypothesis is we would want to, for instance, design drugs around the driver genes, because those, if we could interfere with their activity, maybe the tumor would die or regress. And there's certainly some evidence supporting that. So transitioning to slide 18, we begin to talk a little bit about the genes that contribute to the mutator phenotype. I feel like I've already discussed that quite a bit, but they do describe two particular genes that are involved. And I would just like to stress at this point that this is situations where we're seeing copy numbers of driver mutation alleles increasing in cells over time. And as the numbers of those genes, either through double minute chromosomes, where you see many tiny little DNA circles that have multiple copies of driver mutations on them, or in situations where you've had an expansion, a linear expansion of an array of these driver mutation genes kind of taking over a whole chromosome. The minutes are shown in figure A. These arrays of driver mutations are shown in figure B. We do often see copy number correlating with the type of genomic instability or variation that will build a tumor genome that will allow us to be tumorigenic.